Good morning, everybody. Just a reminder, since we moved to 8:30, we're going to try to start right on time, each time. So welcome. For those of you who don't know me, my name is Damien Rose. I'm one of the co-organizers of the Bay Area Lecture Series. And so we have a couple of different series in grand rounds. The Bay Area Lecture Series is meant to highlight faculty doing great work locally, to showcase them and just think a little bit about collaborations that could happen here as soon as tomorrow. So I want to just briefly introduce Dr. Judy Ford. So I've known Judy since she came here. We were thinking it's around 2007 or 2008. She did her neuroscience PhD and original work at Stanford and UCSF. And so I've really enjoyed sort of being a clinical sort of outsider looking into the work that her lab does. And just a couple of comments, because I think she'll tell you all about it. But what I've been struck by is not only does her work sort of highlight what I would say is a higher order evolutionary system that really makes me think about perception and cognition across animals, and in particular humans, of course, but it also has provided me with a narrative I didn't have before in a way of discussing perceptual disturbance with my patients that I hadn't had before. And it's really helped me build alliance and better understand the experience of my patients. So with that introduction, I'll invite Judy up to tell us about deficits in a basic neural mechanism across psychosis spectrum.
Thank you, Damien, and thank you for the opportunity to talk today about the work we've been doing for almost 2 decades about a basic neural mechanism that enables all animals to discount the sensations that they are experiencing that result from their own actions and allow them to focus on typically more important sensations coming from outside of themselves. Before I really launch into this, I want to give you just a little personal history of how I got here. It was in 1995 at the International Congress on Schizophrenia Research. I emphasize schizophrenia. I was giving my first talk ever on schizophrenia. It was probably on the event-related brain potential, the P300. I was probably saying that it was affected maybe by clinical symptoms or maybe not affected by clinical symptoms. I really don't remember the details. What I do remember, however, as if it happened yesterday, was as I finished my talk, almost finished my talk, a man, stage left, middle row, coat and tie, stood up and raised his hand to ask a question. And I thought to myself, this is a bad start. And he said to me, how can you possibly study schizophrenia? He said, schizophrenia is a heterogeneous disease. He went on to explain to me what heterogeneous means, which I thought was useful. It's got positive symptoms, it's got negative symptoms, it's got disorganization, it's a whole panoply of symptoms. How can you possibly study an illness called schizophrenia? I do not remember if I answered the question. I certainly don't know how I would have answered the question. I do not remember if anyone in the audience who also studied schizophrenia stood up to defend me and help me answer this question. After all, it was a Schizophrenia Congress. But I just have the feeling that I was left dangling there. And at that point made a decision, or shortly thereafter as I was processing, that I wouldn't study schizophrenia anymore. I wouldn't take on the entire illness. I would focus on a symptom. And that symptom would be auditory verbal hallucinations or voices. And as you all know, these are thoughts, inner speech, ruminations coming from inside the self that sound like they're coming from other people or external sources. Okay? So then I could ask myself, all right, now what do I do? How do you study voices with human brain imaging methods? The most obvious and intuitive conceptual way to study voices is to put somebody who hears voices in an MR scanner, or at that point it was actually mostly PET imaging. Put them in a scanner, ask them to press a button every time they hear a voice. Ask them to press another button when they don't hear a voice, because you need a contrast, hope that there are enough such events of both of these types, and then compare. And I thought, that's too hard. And I actually tried it with Ralph Hoffman when we were at Yale, and it was really hard, and I admire the people that do it. One of the things that this method will do for you, though, is it will tell you where in the brain things are happening, where in the brain voices are being experienced. But a simpler way, I thought, would be to study a mechanism by which people might hear voices. And that raises the question, how does the nervous system solve the fundamental problem of distinguishing self from other, or the self-other problem? The answer is the efference copy corollary discharge mechanism, and you're going to be hearing a lot about that. It is a system that has been studied across the animal kingdom. This is just a short list of animals that has been studied in: nematodes, crickets, electric fish, bats, songbirds, rodents, monkeys, and humans. It allows all species to predict and thereby suppress sensations that result from voluntary motor actions and tag these sensations as coming from self. In so doing, self-generated sensations are filtered and processed economically. So I'm just going to give you some examples that I've got here, and I want to I'm going to show you some GIFs in the form of GIFs. I want to thank Josh Woolley for pointing me to the first GIF that was part of David Schneider's Twitter feed about his new article in Neuron, which deals with this subject. He, by the way, and these basic neuroscientists have gotten closer to helping us understand what the real mechanism is of what I call a mechanism. And I also want to thank my 10-year-old son, Adam, who showed me how to download GIFs. Because it's not so simple, if you're me. So, how does the nervous system solve the fundamental problem of distinguishing self from other? That's Schneider et al. Ever wonder why you don't usually hear your own footsteps? Efference copy corollary discharge mechanism. Ever wonder why you can't tickle yourself? Efference copy corollary discharge mechanism. Ever wonder how birds learn to sing like others in the flock? I'll leave this one up because I think it is so amazingly cute. This relates to how humans also learn how to talk like people in their family, people in their hood, people in their culture. The brain automatically detects when there's a slight deviance from what you've produced, makes adjustments, largely unconscious, and you are able to develop a song like your conspecifics, and we're able to develop speech like others in our culture. Ever wonder why you don't usually hear your own thoughts? The efference copy corollary discharge mechanism. Ever wonder why some people, hear voices when no one is talking? A dysfunctional efference copy corollary discharge mechanism. This is the Feinberg hypothesis. Bob Feinberg, in 1978, proposed that a deficit in this basic mechanism could underlie psychotic experiences. If the brain cannot distinguish between self and other, it will have a hard time distinguishing voices coming from inside and voices coming from outside. So, I hope I've convinced you that this is a really cool mechanism, and I just want to show you a little bit about how you might study this in animals. It has been studied, by the way, before I go there, it's been studied in the vestibular system, so how we keep ourselves upright. It's been studied in the saccadic suppression literature, like why is it that when I move my eyes across the room, the whole thing doesn't swim? It's because when I move my eyes, I get a message, my visual cortex gets a message to not pay attention for a millisecond because it's going to look like it's swimming. And what I'm going to talk about today is the vocalization auditory mechanism. So how do we study it in animals? Basically, we record neural activity from the auditory cortex when animals are singing or vocalizing. So birds are singing. And we record neural activity from auditory cortex when the animal is listening to those same sounds played back. More recent studies have also involved recording from the cerebellum and motor cortex, which has given people a much better understanding of the actual mechanism by which this works. The responses to the vocalized sounds during vocalizing and listening are compared to each other. I'm going to show you just one example from the animal literature. It's a bird song example, and I really like this example because it shows us two things about the system. It shows us how auditory cortex is suppressed during singing and how the suppression is precise to the song that the animal thought it was singing. So this neuron right here responds to passive song presentation. This is a paper by Keller and Hanloser, published in Nature in 2009. You'll notice that all of the animal papers published on this topic go into really high-profile journals. The human papers don't seem to get quite the same coverage. Anyway, this paper was written a while ago, and they were recording, most likely, the song of the songbird, playing it back to the bird while listening. And then what you'll notice is this very same neuron stops firing when the bird sings, unless the investigators altered the feedback. So what you do while the bird is singing, you do this little, you perturb the feedback that the bird gets. In fact, you'll see there's a paper by Michael Brainerd and Alison Dope from a long time ago. I think it was a cover of Science, and the picture was a bird with little earphones on. So what they do is they play the song back to the bird, so the bird hears what the bird is singing, but you perturb it, so it's different. And when you perturb it, the neuron fires again. And here, you'll see that the altered feedback is the red line, so there's a big response. The unaltered feedback is the cyan line, just what the bird was expecting, and the red line is the bird was not expecting that, and the neuron respired vigorously. And again, I just want to emphasize that what we compare is the active condition of singing or vocalizing to the passive condition of listening. So how do we study this in humans? Well, we're not too creative, so we just do what they do. We do what the animal people do. We adopt a version of the songbird paradigm. We also, it's the same, there's a whole monkey literature I could have shown you, same sort of thing. What we do is we just adopted a version of that paradigm. But there's some very important differences. Unfortunately, we are not recording from single cells in the brain, typically, but instead scalp EEG. However, we typically get the whole cortex, if you want to think of it that way, but unfortunately we don't get the cerebellum and the thalamus, which I think are very much involved in this system. And we've instructed our subjects to say, they're not freely singing, and they're not making social calls like the birds are and the marmoset monkeys. And we're studying humans on the psychosis spectrum, not wild-type birds, monkeys, and rodents. This is, I think, a cool diagram that I found in a Ramnani paper, a review, Nature review of this system. So the idea is that when you decide to do something, like reach for a coffee cup, you send a motor command to motor cortex, and as a result of that motor command, you've picked up the coffee cup, and you actually have a sensation. You have the cup, it was probably hot, and you pick up on that right away. And what that is, the sensation that you generate is the reafference. It's what you generated. But at the very same time, there is this fast-tracked command that goes parallel. It is a copy of that motor command, and it goes, it's called an efference copy. You're going to be hearing that a lot. It goes through the cerebellar cortex, through the cerebellar nuclei, and it makes a prediction about what you expect to have. So as I reach for my coffee cup, I expect that I'm going to come up in contact with sort of a hard but warm surface. If that happens, all is good. I send a message, or my brain sends a message, I'm totally unaware of it, that what I expected and what happened are identical. No need for further processing. Cerebellum sends that message, says, carry on, you're done. It's a very quick side loop. However, if I reach for that cup of coffee and it's cold, I will send an error message, again, unconsciously, up to through cerebellum up to higher order areas to make adjustments. This is when the brain, the cortex, as we think of it, gets involved. So an error signal is sent up to sensory motor cortex, and it alerts frontal lobes that it needs to make necessary adjustments. This is a more costly process. The very inexpensive, cost-efficient process is the one where the efference copy, where the corollary discharge and the reafference match. So that's sort of a theoretical view of it, and this is also a theoretical view of it, but this puts it into the context of what we do. So my guy has been told to say ah. Let me just say a quick message on why we say ah. Simultaneously, in Germany, Stanford, and UCSF, 3 labs were studying this, and all of us came up with the brilliant idea to say ah. We refer to that as talking. I know it's not really talking, and I apologize for using the word talking, but it's easier than saying vocalizing all the time. So we all said, ah, well, why do we do that? Anybody who's ever tried to record EEG from the scalp knows that you do not want to disturb anything here during the recording. You don't want people to tap the roof of their mouths. You don't really want them to swallow. You don't want them to clench their jaws. All of that really disturbs the recording. So the sound ah, ah, ah minimally affects the EEG. So we all hit on that. So we've told our guy to say ah. He's getting ready to say ah. He has an intention to speak, and he sends that command to primary motor cortex. At the very same time that that motor command is going out down here to make it happen, he has sent an efference copy of that motor plan. And I've come to realize that this efference copy goes from motor cortex, it gets routed through the pons, goes to the cerebellum, comes back up to the thalamus, and probably ultimately gets to auditory cortex, where it deposits a corollary discharge of the expectation. So that would be the expected sound that my guy thinks it's going to sound like when he finally says it. So he says it. Great. And in fact, right at that same moment, I'll go back. At that same moment, it deposits the real sensation of what he said. If they match, if they perfectly match, then auditory cortex response is suppressed. If they don't match, it in humans is also suppressed, but not as suppressed. Here's how we do it. So here's my guy, again, and he's got an EEG. We, of course, have more than one electrode, but for the illustrations here, he's got an electrode on his head, he's got earphones over his ears, he's got a microphone, it goes through a mixer, and goes into the earphones. And he says, this is the EEG coming off that electrode, and he says, ah, 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 like that, okay? And event-related potentials are derived by averaging together the EEG segments time-locked to voice onset during talking, as you can see here in the sort of orangish lines. So that's where the response, the brain's response to the ah, will be seen. The speech is recorded during talking and played back during listening. And the ERPs to voice onset are compared during talking and listening, like I said. That is done in the entire animal literature. And consistent with the animal literature, responses during talking should be suppressed compared to listening, if we are on target with our hypothesis and our paradigm. So what happens? Here's what happens. So here's my guy. You'll notice he doesn't have a cerebellum. This is an old slide. Cerebellum is a more recent addition to my thinking, and the cerebellum will come in later. I apologize for that. It's amazing that you can download pictures of the brain on the internet and there's no cerebellum. What's that about? I know what it's about. Cerebellum is the last great frontier in neuroscience. So here is the ERP that results from all of those segments that I just showed you. Average them all together. This is time on this axis. 0 is when speech onset happens. 100 means 100 milliseconds and on out. This is voltage. Negative is down. This is a negative component. And this is a negative 5 microvolts. And here's when my guy is talking, and here's where he's just passively listening. So what you see is that there's an N100. They call it N100 or N1, 100 because it happens at 100 milliseconds. It's most likely generated in the superior temporal sulcus of auditory cortex, and it reflects activity in the upper cortical layers. And consistent with the operation of the efference copy and corollary discharge mechanisms, N1 to speech sounds during vocalization is reduced. So that's the reduction of N1. You can see in this particular example, it's almost wiped out compared to the N1 to sounds during playback. So during vocalization, auditory cortex responding is suppressed. And again, this is consistent with the large animal literature. This is what I'll be referring to as the N1 suppression effect, or perhaps the corollary discharge effect. We're also interested in what happens before speech. And we have one experiment where the paradigm was set up so that we could actually look at this. Let me just go through this again. So I just showed you what we were looking at before, which was the response of the brain to the onset of a speech. Now we're looking at what happens in the brain right before speech. So right before I say ah. Every time I say ah, something happens. It should, because I've got to get that sound out. So in this example, each trial of EEG is in phase with the other trials. Because I made these data up. By the way, these are not real data. This is just for illustration. And this being in phase from one trial to the other reflects synchrony of neural activity across trials. The EEG, as some of you may know, can be decomposed into different frequency bands, such as theta, alpha, beta, gamma, and they can vary across time. Different EEG bands can be more or less in phase with the stimulus, which is, again, voice onset. And this is what a time frequency analysis produces. This is very different from what you just saw, except that what we have here is time, again. This is 100 milliseconds. This is 0, speech onset. This is 100 milliseconds before speech onset, and that's 200 milliseconds before speech onset. And we were unable to, because of the way the paradigm was set up, we were unable to look at slower activity, but we could look in the beta band and then these two higher gamma bands. So this is a 15 hertz response. And what you see is that the EEG in the beta band, right before I say ah, gets itself lined up. So in every trial, right before I say ah, it gets itself lined up because it's expecting me to say ah. So one of the things we wanted to know was, of course, how does it look during listening? It better be different during listening, or we've got something, some sort of fancy mind reading going on. And it is different. So there is not any of this synchronization right before you hear the sound. There's only synchronization before you say the sound. So we asked, what does this mean? Is this related to anything important? We asked whether it was related perhaps to the suppression that we see, the N1 suppression effect or the corollary discharge. And so we correlated it. We correlated the strength of this signal to the strength of this suppression signal. And we found a good, tight correlation, saying to us that pre-talking synchrony is related to subsequent sensory suppression to the speech sound. And we suggested that it was therefore an instantiation of the efference copy, which is this guy. I'm just going to show you some more data that are consistent with this. This was an analysis done by Jun Wang, who came and stayed with us for a couple of months. He was from UT Southwest. This is our model, and here are the data. He took the EEG data that we had collected. You know, I told you that we had access to the whole cortex. I think we had 64 electrodes over the whole scalp. He was able to do a source analysis using a program called SPM. And what he found was there was a greater activity right here before talking than listening, suggesting that Broca's area is getting ready to say ah. And he found 100 milliseconds after speech onset, consistent again with our N1 latency, that this was greater during listening than talking. In other words, it was suppressed. So this gives us some reassurance that the system is probably involving frontal lobe areas, perhaps Broca's area, and this is superior temporal gyrus. So this is active, and this is not active during talking. This is active during listening, but not active during listen. This is not active during listening here. oops, let's see. Oh, sorry. And he correlated the activity in these two areas with each other and found that the more correlated they were, the more suppressed was the response to the sound. And I have one more thing like this. This is a study we did a long time ago. We were doing but we were also doing real sentences. We were actually having subjects say real sentences, so it's right for me to refer to this as talking. This really is talking. We had subjects say sentences, and then we had recorded the sentences and played them back. And we were looking at the amount of coherence in the EEG between the frontal areas, which we think is where speech is generated, and the temporal areas where the speech is processed. And what these lines, so I said that, so we calculated coherence, and we found that there was more coherence during talking than listening. And this was in the theta band, around 4 hertz. And the fatter the line here, the greater the difference between talking and listening. So talking was always more coherent than listening, and in some areas it was really quite significant, in others barely significant, if you consider 0.05. So this is a strong effect. And interestingly and reassuringly, it was only over the left hemisphere. That doesn't always happen. Again, it would seem that what's happening is there's an efference copy being sent from somewhere in the frontal areas to somewhere in the back of the brain during talking compared to listening. I want to just mention another cool thing about this. I mentioned to you that the bird song has a precision to her song, and if you perturb it and get it a little off, her brain responds differently. Well, so do ours. So this is a slightly different view of the way I've been showing things. What we did in this experiment, we did this at Stanford with Tata, Heinz Maldonado. What we were able to do is perturb the sound. So if you're saying we dial it down half a tone, and it sounds different. And it's perturbed. It's, invalid feedback. It's basically going from a B to a B flat, if you will, if you know anything about music. So you're going down a half a tone. It's not a lot, but it's noticeable. And what you see is this is the playback condition, and the green line is the regular one. The red line would be the perturbed one. Basically, there's no difference during playback. The brain doesn't care during playback, passive listening. It does care while you're talking, however. And if you're talking and you say and you hear all is well. But if you say and you hear it is not so good, and you have a much larger response to it. So the brain is sensitive to the perturbations that we do to the speech sound during talking. And this is one of my favorite experiments ever, because we didn't actually do anything to perturb the sound, but the subjects themselves do something to perturb the sound. This was done by Kevin Setek when he was a research assistant in our lab. He's now a grad student, probably getting toward the end of it, at Harvard and MIT. What he did was he noticed that people, when he was looking at the output of our experiments, he noticed that the ahs are not always the same. In fact, there's a fair amount of variation in how you actually say You think you're saying it the same way, but we're humans, and we make little mistakes. And what he did then was he sorted those trials into the ones that were very much like the previous and the ones that were not at all like the previous So these are the ones that we call the near, and these are the far. And what you see is that when it's just like the previous when your current is just like your previous you have normal suppression. But if it's far off from your previous you don't have so much suppression. And what I like about this is that we did not ask subjects to try to keep a consistent and as far as they know, they did. But the brain knew better. Auditory cortex was on to you and knew that, in fact, it wasn't quite the way it used to be. And we think of this experiment as another illustration of how important this system is to helping us get immediate feedback about how we are doing. And again, I believe it's going to go through some very quick side loop, perhaps through the cerebellum, so that you don't have to really pay attention until something goes a little wrong. So basically what I've shown you so far are some of the data from healthy controls. I want to now focus on schizophrenia. So according to the Feinberg hypothesis, we would expect that people with schizophrenia might have a reduction in this output of the corollary discharge. And in fact, it is reduced in schizophrenia, and that's the amount of reduction in this particular sample. And no matter how hard we tried, we were not able to relate this to hallucinations or delusions, which were our top choices, and we certainly tried other things as well, and it basically wasn't really related to anything. But we persisted, and so we did this at Stanford in a couple samples, did it at Yale in a couple samples. As I'll tell you later, one of our colleagues ran this experiment in Chicago. We have a friend in Australia who's doing this work, and now here at UCSF. So we've replicated this in many, many places, and at the VA. So it doesn't depend on hemisphere, it doesn't depend on coast. It seems to be everywhere that we can find people who are willing to do the experiments. What about the inference copy thing? What happens before speech? This is a bit of good news in our efforts to find relations with symptoms. Here is what happens in people with schizophrenia. So you remember that people with healthy controls synchronized the activity in the beta band before they spoke. People with schizophrenia tend not to. They have less pre-talking synchrony, and again, this would be consistent with the failure of the efference copy being sent from motor cortex to auditory cortex preceding the talking in these patients. In other words, maybe the efference copy is broken. So we asked whether it might be related to symptoms, and it is, modestly. So people who have more symptom severity have less pre-talking synchrony. And this is, again, an EEG map, and it was the EEG in this area of the brain that was most highly related to auditory hallucinations. So again, pre-talking synchrony is reduced in patients, but especially in those who have a propensity towards severe and distressing voices. And you might remember this from a minute and a half ago, where healthy controls show greater coherence between the frontal and the temporal lobes during talking than listening. This is the data for patients. Very little difference between their talking and their listening in preparing to speak. And this is another example where we got a tendency to get a symptom correlation. This is the coherence between talking and listening, and you can see that in patients who hear voices, there is no difference at all between talking and listening. There is a bit of a difference in patients who do not hear voices, and a significant difference in people who are healthy controls. And you might remember this experiment also. We did this in people with schizophrenia. And one of the things that's important to see in this picture, again, is that people with schizophrenia show suppression. So show a lack of suppression. So you can see that just this whole thing is bigger than this whole thing. And you also see that there is not a difference between perturbed speech and unperturbed speech. So the brain in people with schizophrenia who hear voices is less sensitive to perturbed speech. And again, this is work we did at Stanford with Tata, Heinz, Maldonado. So that was schizophrenia. I'm now going to talk a little bit about psychotic bipolar disorder. I mentioned this a second ago, our relationship with Jun Wang. This was our collaborator John Sweeney. He offered graciously, generously to collect data for us in Chicago from a whole cohort of patients that he was studying as part of the BSNP collaboration, which is Bipolar Schizophrenia Network on Intermediate Phenotypes. So he was able to collect data from people with psychotic bipolar disease and schizoaffective disease, as well as just schizophrenia and their relatives. So first thing that you can see here is that this is the degree of suppression. This is healthy controls, and you can see that they have 5 microvolts of suppression, which is a good healthy amount of suppression. So basically we confirmed that effect in healthy controls, which is always nice and reassuring. And we also confirmed that people with schizophrenia had less suppression. But what about psychotic, bipolar, and schizoaffective? They also had less suppression than healthy controls. And I guess it surprised me at the time, but now I don't know why it surprised me, because this is really about psychosis and not about a DSM diagnosis. But it was absolutely the same in schizophrenics, psychotic, bipolars, and schizoaffective. It didn't matter what the DSM diagnosis was. It was about whether they had psychosis or not. Unfortunately, again, this suppression was not related to clinical symptoms, no matter how hard we tried. And I mentioned that John also recorded data from first-degree relatives. So these are first-degree relatives that are unaffected by any psychotic illness. It doesn't mean they don't have anything else going on, but they didn't have psychotic illness. And they were intermediate, so they were, they were in between the probans and the healthy controls. And there was, I think, a linear trend that we reported. So again, some indication that even healthy controls who have a psychotic relative will have some disturbance of this corollary discharge mechanism. Okay, this is something I think everybody here knows a lot about because of the work of Dan Mathalon in the prodrome, in the clinical high risk for psychosis. And this is a picture I think that has been shown everywhere. I'm going to show it again in case somebody hasn't seen it. This is sort of the course of schizophrenia. The red line is healthy controls, and on the y-axis we have functioning. So basically healthy controls set the standard, and they are, of course, at 0. They're healthy, and that's how we define what's normal. People with schizophrenia in the pre-morbid period, before they have schizophrenia, when they're still just toddlers, may perform just a little bit less well as their healthy cohorts. And then there's a period that's called the prodrome, which is when it's sort of leading up to psychosis, where things begin to go a little funny. They begin to have some, begin to have some social problems. They begin to have some unusual thoughts. They begin to hear things that are a little strange, and some of them then go on to become psychotic, and this would be the point at which they're psychotic. This is what we're going to be calling the clinical high-risk people. This would be the early schizophrenia patients, and these are the chronic schizophrenia patients. And what you see in this paper is impress that there are corollary discharge deficits in early illness patients, which we would have guessed, which are here, but there are also significant deficits in the clinical high-risk population, which is here. And here, again, is the healthy controls. I realize this looks more exaggerated than other things I've shown because I tried to squeeze them all together, so it makes it look really big. But you can see that the difference between these two groups is not significant. The suppression in healthy controls is greater than in early schizophrenia or clinical high-risk, who are not different from each other. We then related this deficit to some of the symptoms that the clinical high-risk population expresses. And this is the relationship with the unusual thought content, which is akin to delusions. And you can see that it is a significant relationship. People who have worse suppression down here have more unusual thought content. In large multi-site studies, you'll learn that it's this symptom, unusual thought content, along with suspiciousness, that are the best predictors of who is going to go on to have psychosis, best predictors of impending psychosis. Perceptual aberrations akin to auditory hallucinations are not so predictive. So I'm just going to take a brief detour from this sample. We did another analysis, which I think is important because I think it's sort of helping us figure out how to go forward with all of the data we've been collected. What we decided to do is decompose this N1 into its is the contributions of phase and power. So something that there's no reason for you to know, this is something that only people like us think about, the event-related potential that I've been showing you is actually derived, as you know, from averaging many segments of EEG together. And they represent, the ERP represents possibly, most likely, an increase in power in some of the frequencies, as well as an increase in phase consistency. There's a phase resetting that happens every time you hear a sound. The EEG that was just sort of happily going along, you hear a sound, and the phase is reset. And that adds to how big the event-related potential is, the N1. Also, there's an increase in power. You're just going along, then you hear something, there's an increase in power in certain frequencies. And again, that adds to what we're seeing. So we thought maybe if we pull this thing apart into its constituent parts, maybe we'll get more purchase on trying to relate it to symptoms and even just group differences. So what did we do? By the way, N1 predominantly reflects activity in the 4 hertz band, the theta band. You've heard me say theta before, and I'll be saying theta again. It's in this theta band predominantly that we see the N1 component. So we decomposed the ERP during talk and listen into power and phase consistency. And then we compared N1 suppression to theta phase resetting suppression to theta power suppression. Now, I'm just going to take you through this because I made this slide and I think it's kind of illustrative. This is just an illustration of the phase resetting. So here you are. This is time 1, time 2, time 3, etc. And your EEG is just kind of cruising along, kind of wildly, randomly, not doing much of anything. And then, boom, you hear a sound and your phase gets reset. So in this illustration, you can see that phase is reset every time that sound comes on. This is phase resetting. This contributes a great deal to the events we see on the scalp, as does power. I tried to do a power one like this, but it just didn't work out very well, so I decided to abandon it. So what I'm going to show you right now is the comparison we made between N1 suppression, theta phase resetting suppression, and theta power suppression. These are the suppression measures down here. And this is Cohen's D for the pairwise group differences against healthy controls. So the first thing you see is what I just showed you before, which is the N1 suppression in the clinical high-risk group and the early schizophrenia group. And you can see that the Cohen's D is substantial in the early schiz, and it's okay in the clinical high-risk population. But this is what I just showed you, just with the additional information about Cohen's D. But this is what is striking to me, is that when we look at the theta phase resetting, this is a much better way of distinguishing between groups, certainly between healthy controls and schizophrenia patients. And indeed, this is hot off the press. It's something we're still writing up. It is related to delusion severity. You might remember that in the clinical high risk, delusion severity was related to N1 suppression. It is not in this sample related to theta suppression, but it is in the early schizophrenia patients. It is related to theta phase suppression. Okay, I'm going to move on to schizotypy. Probably everyone knows what schizotypy is, but for those who don't, I'll run through it real quickly. This is our colleague Tom Whitford, who has his own lab now at the University of New South Wales, and he's doing these same kinds of things. He's studying the corollary discharge down there. And he had he has access to a large undergraduate population, and he gave them the schizotypal personality questionnaire, and this is where you say how severe these experiences are for you. Do you have unusual perceptual experiences, suspiciousness, odd beliefs or magical thinking, ideas of reference, excessive social anxiety, no close friends, constricted affect, odd or eccentric behavior, and odd speech? Kind of like psychosis, but not quite like psychosis. And what he showed was that these were the people who had low schizotypy. These are the undergraduates who don't have a lot of that, and these are the undergraduates who have a lot of this. And you can see that there's less N1 suppression or a corollary discharge deficit in people, even with schizotypy. One of the nice things about this is these people are, of course, not medicated. So if you think medication is a problem, this would suggest that not in this sample anyway. And this was related to the severity of schizotypy. Targeted auditory training. I think most people here know about that too. When we first got here in 2007, we hooked up with Sophia Vinogradov, who was doing Mike Merznick's Posit Science. And so we decided we would study these people who were getting targeted auditory training. There were two things of note in our auditory training. One was in the sort of speech perception domain, so they were training people to hear sounds in the speech spectrum better, and also doing a verbal memory training. And what we found, so the question was, can targeted auditory training improve corollary discharge function? And we compared them to a computer games group. So 40 hours of targeted auditory training significantly improved the corollary discharge function, but not up to the healthy control values, which would be up here at 0. These are Z-scores against healthy controls. 40 hours of computer games marginally worsened it, 0.08 level of significance. I think one of the things that's interesting about this is really due to the effect of talking. So it was really during talking that the targeted auditory training seemed to improve the corollary discharge, not during playback. So this is the talking data. So again, I think that the targeted auditory training may have preserved the brain's or restored the brain's ability to suppress sensations coming from self over coming from external sources. Let me see. Okay, finally is our ketamine data. This is our the glutamate model of psychosis. As probably many of you know, the NMDA receptor hypofunction has been implicated in the pathophysiology of schizophrenia, and it may, in fact, underlie corollary discharge deficits. So what we did was this was at Yale, where you can do these studies. We had healthy controls. This is when they got an infusion of saline, and this is when they got an infusion of ketamine. And what you'll see is that these data, the ketamine data, look a lot like the data we recorded from patients with schizophrenia. We did a statistical test. We did a Z-score test comparing the patients to healthy controls and comparing the ketamine to the placebo, and there was no statistical difference between schizophrenia and ketamine, which might make you think that schizophrenia is like ketamine in a bottle, or yeah, ketamine is like schizophrenia. But this is what's interesting about this is all the new data about ketamine helping depression. And so I called Toby Martone yesterday to find out what the doses are here. They're the same. They're basically the same. So we produce schizophrenia and they produce wellness. Go figure. Of course, it goes away. So back to those big questions. So let's remember my man, stage left, middle row, coat and tie, wanting me to think about symptoms and what is it about, symptoms. So I'm asking the question, clearly, I think efferent's copy and corollary discharge are affected by psychosis. You see differences from clinical high risk and schizotypy, with even intermediate effects in unaffected family members, all the way up to psychotic bipolar patients, first episode patients, chronic schizophrenia patients. It's even reduced in a glutamate model of schizophrenia. So what is it about psychosis that is responsible for these effects? Is it just vulnerability? It doesn't seem to be symptoms. It is symptoms, but only to a modest extent, and maybe we aren't quite there yet. And I know the man in the middle row, stage left, would not like this, but maybe it's possible that we have to consider the possibility that psychosis breeds truer than the symptoms it comprises. But I'm not ready to give up on that yet. I think we also have to consider other possibilities. I think our hot-off-the-press data where we decomposed the ERP into phase and power might be an avenue forward. I'd like to take some of the data that were disappointing in the past and see if we just pull it apart, might we begin to find relationships that we had given up on. Also, I really want to start focusing on the cerebellum. As I pointed out, this is my old picture. This is my new picture. I did have a cool GIF of the final frontier, but it just wasn't working that well, so I threw it away in the last minute. But I think the cerebellum is really where a lot of this is happening. I just have to figure out how to get purchase on it and how I can do things with the cerebellum. We're thinking about doing electrical and magnetic stimulation of the cerebellum during these kinds of experiments and seeing whether we can move it in some way. There's a lot of work now in the animal literature on cerebellum and the corollary discharge, and I've begun to have communications with some of these people, so I'm hoping to move this forward in that way. Also, maybe we should be asking about agency and not hallucinations. A lot of this is about agency. You know, when you move, you touch something, I am the agent of this feeling. I caused this feeling. And so we do have agency questionnaires that we are now going to begin to ask patients to fill out, and whether they have a sense of lack of agency. I did mention medication. Most of these people are medicated. The clinical high-risk patients are, by and large, not medicated, and the schizotypic patients are not medicated. And certainly the first-degree relatives, if they're medicated, they're not medicated for psychotic symptoms. Also, I think we really need to take advantage about what is known from studies of animals and how easy it is to study this in animals. For example, we're actively teaching mice to say, just kidding. Instead, we have colleagues who are teaching mice to press a button to hear an aw. And this paper was published a while ago. There's another group, the Snyder group that I mentioned, who was responsible for that GIF of the foot walking. Thank you, Josh. You missed my call out to you. So there are people doing this, push a button, get a tone. There are people doing movement while hearing tones. Schneider is one of the people that's doing that. And so is Torfi Sigurdsson. And so instead of teaching mice to talk, we're teaching humans to press a button to get a tone. And we have a whole bunch of data that are waiting to be analyzed on this. We published one paper showing the same type of effect where healthy controls show suppression of auditory cortex when they push a button to hear the tone compared to people's schizophrenia. But with that, I'm going to wrap up, and my final GIF. And want to read this quote from Mark Sommer and Trinity Crapes, who is very committed to the corollary discharge system. He says, or they say, in addition to the usual flow of information from sensory systems to motor systems, there is extensive signaling in the opposite direction by motor systems reporting their activities to sensory structures. It is this coordination between the two systems that makes it possible to analyze the world while moving within it. And with that, I want to thank a whole bunch of people who have helped put this work out, from colleagues at Stanford, Yale, UCSF, and again, Australia and the University of Illinois University. And, of course, the funding agencies, the VA, NARSAD, NCRR, and grants from NIMH. And now it's your turn. Are there any men in the middle row who want to ask an aggressive question? Okay. I understand we have 5.5 minutes. So, hi, Craig.
This is a difficult question to ask, but let me try. I can understand why you want to look at the correlation or association with things like delusions and hallucination symptoms of schizophrenia,
right,
because if you get too far removed from schizophrenia, then you don't know whether it's related, and of course that's one of your aims.
Right.
On the other hand, you think, and I'm sure you do, that there are going to be, 15 to 300
That's right.
in between your process and actually having delusions and hallucinations characteristic of schizophrenia. So the question is there something that's kind of closer to what you're doing that's more measurable, objective? And let me give you an example. There must be lots of examples. That executive functioning tends to be perturbed across psychosis, and unrelated to whether you have schizophrenia, bipolar, psychotic depression. Would something like that be a factor or a correlate? You mentioned idiosyncratic thinking as being associated. Now that's kind of interesting because that's a little bit more related to schizophrenia in a way. But anyway, you must have been thinking about this. You might get closer to basic behavioral phenomena.
Yeah, no, I appreciate that question, and I think that's part of the reason I think we should be looking at agency instead of voices, because I think voices, first of all, voices I think are hard to study. I think people who hear voices don't always have a clear sense that they're hearing voices. Often I could go on and on about voices, but something more like agency might be a little bit more objective and tractable. Your comment about cognitive function, I think that, first of all, we've looked at that, and it doesn't relate. And I don't think it will, because I think this is something that is it's dependent on a very subcortical, quick side loop that the cerebellum is involved in. I don't think it's going to have cognitive overtones. The fact that you see this mechanism in nematodes, which only have 302 cells, and probably I don't know whether their working memory is normal or not, but I think that it's too basic and elemental to be thinking that cognitive dysfunction might be related to it. But believe me, when we have measures of cognitive dysfunction, and I try that to see if it's related to this also. And so far that has not come up. But there are a lot of other things that go into psychosis, at least chronic psychosis, that could be contributing. But again, because we've got the data from the clinical high risk, who have not already had a lot of the bad, sequelae of the illness, they aren't living in tents, they don't have, their lives are not yet as impoverished, and we still see it there. What I ultimately come down to is I think it's a risk factor for psychosis. I think it's a vulnerability for psychosis. But what about psychosis? And that's the problem. I think it is vulnerability, but I don't know what exactly it is about psychosis. I don't think I've satisfied you.
I'm just thinking about it.
Okay, we can continue this. Yeah, hi Descartes. Yeah. So probably, and also I want to point out something. Thank you for raising that, although it's only a little tangentially related. This is a system that actually can also augment sensory processing. This is a system that you're getting feedback about how you're doing, so that you can improve your processing. Certainly, people who are blind and are reading Braille, they don't want their sensory system to be suppressed when they are moving their fingers. They want it to be hypersensitive. So it goes both ways, it certainly goes both ways in humans. It may not be so bi-relational in the lower animals. Yeah.
My question was, I think you described two different pathways basically for the first copy. One of them was this frontal oral pathway, cortical pathway. It's fairly specific. But then you also mentioned earlier, there's this cortical subcortical pathway that involves the pons brainstem area. Do you think that there's a distinct function between those two pathways?
I have to, the whole cerebellar, the whole motor, cortex, pons, cerebellar, thalamus, cortex loop is, I think that has got to be part of what we're seeing when we see our correlations, our coherence between frontal and temporal. But the EEG, we are not able to see what's going on in cerebellum or thalamus. I think what we're seeing is the end result of that more elaborate route that I had go through the intention to pons, to cerebellum, the thalamus, and back to cortex. I think that what I'm seeing with that is the end result of that pathway. Yeah.
My question was about depression. Thinking about this discharge mechanism you're talking about, is it at all related to the ataxia that one experiences with depression? I don't know what the neuromechanism of that is, but it sounds like it might be cerebellar.
Interesting. Certainly, when I start, I started being very interested in cerebellum, and ataxia often comes up when I put in the search terms cerebellum and corollary discharge. Ataxia pops is right up there at the top. It certainly could be.
I wonder if that's correlated at all with the psychosis that one
Yeah.
severe depression.
I do not know if John Sweeney, when he was, first of all, his bipolar patients weren't necessarily depressed. But that's, we are now moving into, with Susanna Fryer, we are now taking the giant step into depression work. And once we've got them in there, we're doing a different study, but once we've got them in there, we could certainly be asking those questions and recording these data again. It's a good idea. Thanks. Yeah.
Do we know how suppression varies with more like natural images each? Like the other thing, I was wondering whether this mechanism is
Yeah. Yeah, that's a good question. I'll just, on the first question, just anecdotally, sometimes Brian Roach, who is a first author on a number of these papers and works with us, has noticed that he thinks that now that we're having people talk for a long time, we have long experiments, sort of toward the end of the experiment, things seem to be getting better. But that's just his, that's absolutely just an impression he's gotten. What was the other question? Oh, thinking to oneself. So Tom Whitford, the guy in Australia, is doing that, has done that, and has published it. And yes, he sees suppression related to the thought, the inner speech, that is specific to the kind of sound that the subject is producing. So yeah, you can just go into PubMed and look for Tom Whitford, T.J. Whitford, inner speech, and you'll get that paper up. We weren't part of that, but he told us about it.
I'd like to thank Judy one more time. I know there's lots more questions. I have one, but I will let those stay to where they need to be. Take care.
Thank you, David.